this question on protein synthesis. And if you remember, Alia, that there was this another student who was asking a question that, Miss, why are we reading this strand, the lower strand? Why are we reading this strand? And why are we not reading this strand? Because in this diagram, there is no indication given to us that any of the strand is like which of the strand is 5 prime to 3 prime and which is 3 prime to 5 prime. So you tell me what could be the possible answer to this question? कि कैसे हमें पता चलता है कि we have to read this strand and we are not supposed to read this strand? Any idea?
Miss, I'm not really sure about this.
It's okay, never mind. Now see, the hint is in the question. Figure 4.2 shows first 7 amino acids of the beta chain of hemoglobin. ठीक. The first amino acid in the sequence is valine. The 21 base pairs in the sequence of DNA that code for these 7 amino acids. Acha. First valine hai. Baki ka nahi pata. Baki, of course, they have left it to us so that we can decide. So if we very quickly go through this table to see कि valine का कौन सा code है, right? So we will see that valine has the code GTT, GTC, GTA, and GTG. ठीक है? If I just hold my device like this, so if we look at कि इसमें से कौन सा यहाँ पे है, सारे यही हैं GTT, GTC, GTA, and GTG. तो उनमें से सिर्फ GTG is over here. ठीक है? Since we have done the features of the genetic code and we said that one of the features or salient features of genetic code is that it is redundant. Redundant का मतलब ये है कि more than one triplet of bases can code for the same amino acid. ठीक है? इनमें से जो यहाँ पे मौजूद है वो GTG है. Since it's GTG, so then this means this strand is the readable strand. This strand is not the readable strand. Are you getting this?
Yes, Miss.
समझ आई बात? एंड देन अच्छा आगे की बात तो समझ आ गई थी ना, और यू वर नॉट एबल टू अंडरस्टैंड. इनफैक्ट ऐसे करती हूँ कि आई विल रिपीट इट. नाउ द एग्जामिनर इज सेइंग यूजिंग फिगर 4.2 यानी ये वाला फिगर and table 4.1, use these two things, this one and this one, to state what will happen to the sequence of the amino acids in the first part of the beta chain if the base pair at position 6 is deleted. So this is position 6 and this is the base pair which is deleted. If we remove this base pair, then our reading frame will become, ये तो नहीं change होगा, GTG CAC. ये निकाल दिया हमने बीच में से, तो I'm going to read it as CAC. तो चलो CAC तो पहले ही था and it was coding for histidine, तो histidine तो बन जाएगा, right? But then TGA अब बन जाएगा next code. And TGA जो है ना, if we look at TGA, it is corresponding to a stop codon. ठीक है? So in this case, what will happen? Valine and histidine will be made, but after that there will be no protein made. Can you just excuse me for a minute, huh?
Then, um, in the third box, won't the GC base take over? And won't it be substituted if that base is deleted?
Which third box?
Miss, the seventh base pair.
7 base pair, तो वही तो हम यूज कर रहे हैं. देखो CA, जस्ट फॉरगेट कि कोई बेस पेयर डिलीट हुआ है. CAC अगर होगा तो इट विल मेक हिस्टिडीन. ठीक है? यू रिमूव G एंड C. तो नाउ लेट्स कंटिन्यू. CAC, ये एक दूसरे से मिल गए. CA और C, इमेजिन करो कि बीच वाला C नहीं है, ये वाला C है, तो CAC है तो हिस्टिडीन बन जाएगा. सो नाउ आवर नेक्स्ट कोड विल स्टार्ट फ्रॉम TGA. ठीक है? Did you
But
understand
then, Miss,
this?
how will it only be two amino acids long?
2 amino acid long. Why? Because valine and histidine will be made and then it is followed by TGA. So TGA is coding for a stop codon. Stop codon means no amino acid will be synthesized. So when no amino acid is synthesized, तो यहाँ पे polypeptide chain की synthesis रुक जाएगी. Are you getting the point? It will stop.
Okay, miss.
Yes, and when we were this because this concept is related to the concept of the salient features of the genetic code. One of the salient features of the genetic code was that it is punctuated. Remember हमने salient features किए थे, हम दोबारा से कर लेते हैं उसको. That the salient features of the genetic code is that it is punctuated. Look, number one was that it is a triplet code. ठीक है. Second क्या था? It is a universal code. यानी कि सारे organisms में यही 4 bases हैं. And the same triplets code for the same amino acids in every organism. अच्छा, उसके बाद it's a degenerate code or it's a redundant code. It's a non-overlapping code. Non-overlapping का मतलब कि हम जैसे पढ़ रहे थे ना उसको CAC, TGA, CCG. We will read it in triplets of bases. We can't read it like this कि पहला मैं ऐसे पढ़ूं CAC और second मैं पढ़ूं ACT and third मैं पढ़ूं CTG and so on. It's a non-overlapping code and it's a punctuating code. It's a punctuating code means that it has a stop and a start codon. तो जहाँ पे stop हो जाएगा वो stop हो जाएगा, खत्म. Right? Acha, so let's go to this. Once again, the question. Huh. Now the next one. What will happen if the 3 base pairs at position 7, 8 and 9 are deleted? यानी 7 वाला base pair भी deleted, 8 वाला भी and 9 वाला भी. 7 को देख लेते हैं. 1, 2, 3, 4, 5, 6, 7. अच्छा, I am deleting this base pair. 7, 8, 9. तो इसका मतलब हुआ कि 7, ये पोजीशन 7 है, ये 8 है, ये 9 है. So this means I'm deleting the entire triplet of bases. So if I delete CTG, they are asking what will happen. CTG stands for what? यहाँ पे देखो. Look at this and tell me CTG stands for what?
Miss leucine.
Leucine. तो इसका मतलब leucine will not be made. Right? Leucine will not be made, but the rest of the amino acids will be made. क्यों? इसलिए कि कोई स्टॉप कोडॉन नहीं है आगे. Are you getting the point? बिल्कुल ऐसे जैसे कि आपके पास ये कोड बिल्कुल इंटैक्ट है. The cell will make valine. This code is intact. The cell will make histidine. This is completely removed. The cell will not make leucine. This is there. ACT. What is ACT coding for? Look at the table and tell me.
Miss threonine.
Threonine बन जाएगा और उसके आगे जो भी बनना होगा वो बन जाएगा. So the answer is that here all 3 bases are deleted, so that particular amino acid is not made, which is leucine. Right?
Yes.
ठीक है, आगे करते हैं. Let's do this. Let's do the revision of this. DNA is involved in the processes of replication and transcription. Complete figure 4.2. Complete table 4.2 by using a tick to indicate which features apply to each of the processes and use a cross for features that do not apply. हाँ जी.
Miss hydrogen bonds are broken in both of them.
Hydrogen bonds are broken in both of them. Okay, whatever you are saying, I'm just putting a tick and a cross, and then the end we'll look at the. One second. Achha, second.
Miss both stands for replication and not for transcription.
Both.
For replication.
Achha, okay. So there'll be a tick over here and cross over here.
Yes.
Achha. Third one.
Miss both of them.
Phosphodiester bonds are formed. Okay. Last one.
Miss replication.
The the answers are not given. Were we right? We were right?
Yes, miss.
Let's look at the other questions. 4-mark question. We haven't done telomeres. Telomeres to mitosis mein karenge. Let's suppose I am asking this question. Describe the function of ribosomes in protein synthesis. You just tell me the points जो भी आपके दिमाग में आते हैं, and I'll keep on writing. ठीक है?
Miss, ribosomes are used in the transcription part.
No.
I mean the translation part, miss.
Okay, but it says describe and it's asking for 4 points.
Miss, in the tRNA molecule, the amino acids are brought together and the phosphodiester bonds are formed.
But what is the connection of this with the ribosome?
Because miss, this all happens in the ribosome. The mRNA is held there and the tRNA passes through it.
Okay, so then in
to
this
form a
case,
chain.
wait, now you're changing your sentence. So I'm changing it, and now you tell me the exact thing to write down. Now you will see, since you are telling me your point, so you will realize what mistakes you are doing, right? Whatever you write down, you have to make sense of it to the question that is being asked. So जो भी आप कह रहे हो, उसका connection बनाओ function of ribosomes के साथ. So yes, now change your sentence. What happens to tRNA? Oh, sorry. And what is the connection of tRNA with? Can you see my
Miss,
screen?
can I try?
Sorry. Yes, Amna, are you there?
Yes, Miss, can I try?
Yes, you
Answering
can
the
try.
question.
Just a minute. Let me. Can you guys see my screen?
It's not right now.
Acha, okay. Suddenly, I don't know what happened. Just a minute. Yes, you can try, Amna.
Mr. Ribosome
Hmm.
ribosome helps with translating the genetic code written in the transcribed in the mRNA and turns it into an amino acid sequence.
But how? They're asking you how. Describe means how. How does it
energy from respiration?
Energy from respiration. Okay, to do what?
So translate the code.
Translate the code is a very general and a vague answer. But to actually talk about what is that thing in which the energy in the form of ATP is needed to power a certain specific process in translation or in protein synthesis. In order to form a polypeptide chain, we need peptide bond formation between two amino acids, and this happens through the action of the enzyme peptidyl transferase. Let's go back to our slides where we were talking about the function of the like whatever the ribosome was doing, and हमें खुद ही दिमाग में ख्याल आ जाएगा कि राइबोसोम कर क्या रहा था, right? So basically we say that huh, we say that the machinery that is needed for translation is actually ribosomes. And of course, tRNA and mRNA, right? So look at this. The mRNA is binding to the small unit of the ribosome. जब आप राइबोसोम की बात करोगे तो आप स्मॉल यूनिट के बारे में अलग से बताओगे, लार्ज यूनिट के बारे में अलग से बताओगे. So read this. The mRNA, I'm highlighting it, the mRNA is binding to the small unit of the ribosome. 6 bases at a time are exposed to the large subunit. Okay? So what is happening? The mRNA is binding to the small subunit and 6 bases at a time are exposed to the large subunit. So now this specific point जो है ना ये आपको बताती है कि actually ribosome का direct role क्या है protein synthesis में. अगर आप ऐसे generally बोलोगे ना कि ribosome is helping in the translation of that code that was transcribed in transcription, that's a very vague answer. तो क्या हो रहा है? mRNA is binding to the small and 6 bases. 1, 2, 3, 4, 5, 6. 6 bases are exposed at one time to the large subunit. Are you understanding this thing?
Yes.
Right? अच्छा. And then another point. ये तो आपका इसमें से हम कह सकते हैं कि this can be one point and this can be a second point. And a third point is that each tRNA molecule with its specific amino acid brings the amino acid to the ribosome. So this tRNA जो आपको नजर आ रहा है, जो ऊपर अपने साथ एक amino acid उठा के ला रहा है, where is it coming? It's actually coming and bringing the correct amino acid to the ribosome. So this can be a third function. This question was carrying 4 marks. ठीक है? Let's see. The tRNA docks at the binding site in the ribosome. Isn't it docking? Look at the tRNA. ऊपर थोड़ी देर पहले यहाँ था. For example, it's over here and it's docking. It's docking at a binding site. ये इसका binding site होगा, right? In the ribosome. So look at these specific functions of the ribosome, right? Now, if we go back to that question, जहाँ हम थे, and we can also go through the mark scheme to see the exact function of a ribosome. So, this is what, just a minute. The question was this one. It is part E, okay? So let's look at the mark scheme of part E. Okay. Have a look. Some of the points, like for example, Amna and Alia, both of you, you did mention this point. ठीक है, translation तो करता है. Even if you mention a general or a non-specific point like this, maybe you can get 1 credit. Okay? But what about the other marks in which the examiner is asking you to describe? इसका मतलब वो आपसे ये कह रहा है, how is this taking place? How? Describe means how and explain means why. तो how में मतलब आपको उसकी details बतानी है, हो कैसे रहा है. So 1 mark you'll get and then you are going to talk very specific कि small unit जो है वो attach करता है, it's a binding site. And then also provides binding sites for 2 tRNA molecules. 2 amino acids are held close together. There is peptide bond formation that is taking place. ये सब कुछ राइबोसोम के ऊपर ही हो रहा है through the action of this enzyme peptidyl transferase. And the examiner says alternate valid point is also acceptable. तो वो slide है ना वहाँ पे उसमें और भी बहुत सारी alternate valid points थी. तो I would like both of you, Amna and Alia, that you should note down these points. In fact, हम अपना आंसर खुद बनाते हैं, ठीक है? Let's make our answer ourselves. So let's suppose if this is the question, I would say first point would be, and please note note this point down. When you're done noting it down, do let me know.
Sample.
Alia, what about you, beta?
Yes, Miss Sam.
Okay, done. Let's do some more questions. I have lots of confusing questions over here. Okay. Let's suppose I ask you this question. Use figure 4.1 to explain how the structure of mRNA differs from the structure of DNA. In the picture, what can you see? Can you see DNA or mRNA? Yes, girls, is it DNA or is it mRNA?
Miss, it's DNA.
It's DNA. What tells you it's DNA, beta? Alia?
Miss, because there are two strands, anti-parallel.
Correct. Two polynucleotide strand. Acha. How many nucleotides can you see in this picture? If I ask you about like the entire molecule, how many nucleotides can you see in this picture?
Miss, 6 nucleotides.
Correct. How many bases? In this picture only. 4 only, isn't it? 4 ही
Yes,
नजर
Miss.
आ रहे हैं. हाँ, हालांकि 1 तो 6 चाहिए लेकिन वो दिखाया नहीं उन्होंने. अच्छा. And then, what is the name of? I think we'll stop here because the meeting is going to end. So I'm restarting the meeting because we are not on LMS right now, right? So I'm ending and I'm restarting.